Rabies virus by Joshua Sherman. The history of rabies virus dates back to the 1st century AD, when the first Roman scholar named Aulus Cornelius Celsus suggested that rabies was transmitted by the saliva of the biting animal. Celsus incorrectly suggested that a cure for rabies would be holding a victim underwater. Those that didn't eventually drown from rabies died from it. Other suggested cures were burning the wounds with a hot poker and ingesting the hair of the animal. In addition, hairs of the animal were laid on the wound. Rabies has been relevant for hundreds of years and has been responsible for multiple epidemics. In 1997, there was a canine rabies outbreak which resulted in at least 113 deaths in Flores, Indonesia. About 50% of dog bite cases and human deaths were in children under 15 years old. Between 1998 and the end of 2002, there were 3,389 cases of rabies that were all treated successfully. In addition to the Flores epidemic, another severe epidemic happened in the Republic of Angola, where rabies claimed the lives of 93 children in 3 months in the country's capital city, Luanda. Once the country had gotten a vaccination and post-exposure treatments, they were able to save many lives. In the United States, approximately 5,000 animal rabies cases are reported annually to the Centers for Disease Control and Prevention, otherwise known as the CDC, and more than 90% of those cases occur in wildlife. The number of rabies-related human deaths in the U.S. has dramatically declined since the early 1900s. Then, more than 100 annually died compared to 1 or 2 per year since 1960. This major decline in deaths is attributed to successful pet vaccination and animal control programs, public health surveillance and testing, and availability of post-exposure prophylaxis for rabies. Biology of the virus. Rabies is a dangerous viral disease that is fatal when untreated. Rabies is transmitted through the bite of a rabid animal, and it affects the central nervous system of mammals, ultimately causing disease in the brain and then death. The structure of rabies virus is quite interesting. This disease is classified in the Rhabdoviridae family due to its distinct bullet shape. Rhabdoviruses are usually 180 nanometers long to 75 nanometers wide. The genome encodes for 5 different proteins: nucleoprotein, which is N, phosphoprotein, P, matrix protein, M, glycoprotein, G, and polymerase, L. There are 2 major structural components, which are the helical ribonucleoprotein core, also known as RNP, and a surrounding envelope. Rabies virus is a negative strand RNA virus, and it enters a cell via membrane fusion. The virus attaches to a host cell via the G protein spikes, then penetrates into the cytoplasm by fusion, and the RNP core is uncoated. The core initiates primary transcription of the 5 complementary messenger RNAs by using RNA polymerase. Each RNA becomes translated into an individual viral protein. After viral proteins have been synthesized, replication of the genomic RNA continues with the synthesis of full-length positive stranded RNA, which acts as a template for the production of more negatively stranded RNA. After being infected with rabies virus, it may take between weeks and months before symptoms have appeared. The incubation period of the disease is dependent on the location of the exposure site and how far away it is from the brain. The first symptoms that appear are similar to those of the flu, which include weakness, fever, and headache. The acute period lasts between 2 and 10 days, and clinical signs of rabies include cerebral dysfunction, anxiety, confusion, hallucinations, hydrophobia, insomnia, and more. Once these signs appear, and if not treated, rabies is almost 100% fatal. In order to avoid getting rabies, there is a pre-exposure vaccination. Candidates for pre-exposure vaccinations typically work in laboratory settings and are animal control and wildlife officers. The primary vaccination is 3 1 milliliter injections of HDCV, human diploid cell vaccine, or PCEC. purified chick embryo cell vaccine, which is administered in the deltoid area. There is 1 injection per day on days 0, 7, and 21 or 28. If someone does become infected with rabies virus, they should immediately get post-exposure prophylaxis, or PEP. This treatment consists of a dose of human rabies immune globulin, which is also known as HRIG, and should be given on the day of the rabies exposure. A dose should be given again on days 3, 7, and 14. To create preventative measures of the rabies virus, all animals and pets should be kept up to date on their vaccinations. This way, animals can live safe lives rabies-free in addition to humans as well.